This is the first of a 4-video series where we're going to look at the basics of nitrogen metabolism. In this video, we're going to view nitrogen metabolism from sort of 30,000 feet, think about the general principles. And then in the next video, we're going to talk about the various chemical moves and molecules involved in human nitrogen metabolism. And then we're going to go into specific discussion of amino acids and nucleic acids. So I guess let's launch this thing. So nitrogen metabolism from a sort of high level view. A really good place to start is thinking about the different classes of biomolecules that we have. So metabolism is really built around these 4 types: fats, carbohydrates, amino acids, and nucleic acids. And the big break that happens here is between carbohydrates and amino acids, right? That's where you're adding amino groups. And if you just think about the different biological roles of amino acids and nucleic acids, all kinds of interesting things can happen for them that really can't happen when you're down at, say, a carbohydrate, right? You can get all kinds of very specific hydrogen bond patterns. You can prime to do specific chemistry. You can use things as like a base and an active site, for example, all because of the addition of these nitrogen moieties everywhere. These give new hydrogen bonding potentials, they give new chemistry potentials, they give new structural potentials. The peptide bond involves one of these nitrogens. So it's really a big deal that we can work with nitrogen. And understanding biochemistry really means we have to get a grip on at least some of the nitrogen chemistry that's possible. Okay, so that's our motivation. We want to understand nitrogen chemistry in biomolecules. Let's start looking for patterns. So here I'm showing a nucleic acid and 3 amino acids. And what I'd like you to do is look at this for a second and ask yourself, what's common about these nitrogens? What unifies these different nitrogens? So maybe pause for a second and think about it. What's common about these nitrogen atoms? Pause. Okay, I'm assuming you paused and thought about it. The thing that I notice is that they are all reduced. We don't see any nitrogens bound to oxygen. We just see a bunch of nitrogen atoms bound to either carbon or hydrogen. And basically, if you look across all amino acids, nucleic acids, and basically all cofactors, many of which are derived from, say, amino acids, they're basically in their reduced form. And so they are ammonia, not nitrate. And this is a little bit weird. And to help illustrate how weird it is, let's actually look at the rest of the CHNOPS atoms. So I know this is a big wall of things, but hopefully it makes sense as I walk through it. So what I've done is I made a table with all 6 of the CHNOPS atoms: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And then what I'm showing here in this sort of column is the material that we take in as humans. So one of the ways we take in carbon is as glucose. One of the ways we take in hydrogen is water. We take in nitrogen via amino acids. We take in molecular oxygen via oxygen. We take in phosphorus via phosphates. And we take in sulfur via things like cysteine amino acids. Okay, so this isn't, obviously we take them in different forms too, but this is sort of a representative form. For example, this is one of the ways we take in carbon. How do we put that element out? Well, carbon is actually pretty simple. If we do any metabolism on it, we put it into the, say, central carbon metabolism, it's going to come out as carbon dioxide. That's the way things leave. It's the way carbon leaves our body. So we're going to take it from glucose to carbon dioxide. Water gets basically left alone. An amino acid is going to get turned into urea. Molecular oxygen is going to turn into, say, water. Phosphate stays phosphate. And then something like a sulfide on a cysteine is going to turn into a sulfate. And you're like, okay, wow, that was a big table. How does that help us understand nitrogen chemistry? Well, let's add one more layer of information, and that is, let's ask what happens to the oxidation number of the particular element over the course of metabolism. So we take in sugar, glucose. It has an average oxidation number for the carbons of 1. And we put it out as 4. We have oxidized this, right? We've made carbon dioxide. That's paired with, say, molecular oxygen. So molecular oxygen comes in with an oxidation number of 0, leaves with it of -2, and so we have reduced this, right? We can start playing this game for all of the other atoms. So let's go down here to sulfur. What we see is it goes from an oxidation number of -1 to +6, which means it is oxidized. Water, as I've written it, doesn't change, so stays the same. It stays reduced. Phosphate stays reduced. Sorry, stays oxidized. I said that backwards. These stay oxidized. And nitrogen's weird. It stays reduced. In fact, if you look across the rest of metabolism, you either see things that look like this, where you are oxidizing something that started reduced, or you have things like this, where you are keeping something oxidized that was already oxidized. The weirdo is nitrogen, which you leave in the reduced form. And so we can summarize this at kind of a high level that in general, organisms tend to oxidize carbon, hydrogen, and sulfur. At least they'll oxidize sulfur, hydrogen, if they have some reduced hydrogen around. They will reduce oxygen. They will go from O2 to H2O, and that's going to release energy. So together, these release energy. They're going to leave phosphorus fully oxidized, but then they're going to leave nitrogen fully reduced. And there's this question of why? Why is it kind of the outlier? Why is it doing a different sort of chemistry? And the reason that an organism might want to do this is because it actually would release energy. It leaves energy on the table. So what I'm showing here is the standard state reduction potential of going from nitrate to ammonia. And then the standard state reduction potential of going from oxygen to water. And what you'll notice is this number is bigger than this number. That means electrons will tend to flow in this direction. So if you write out a balanced redox reaction for this, what you actually get is that you would yield 384 kilojoules per mole of ammonia that you put through this process. So it would give you energy if you could burn ammonia into nitrate. Why so organisms do this, right? Organisms are big fans of extracting energy from the environment. What is the limit that makes it so this reaction doesn't happen? And there's a couple. One of the limits is probably that life evolved in a reducing environment. So when we think about the core pieces of metabolism, things like amino acids, nucleic acids, and the like, they actually evolved when there wasn't a bunch of free oxygen around. In fact, there was a fairly catastrophic event where cyanobacteria started producing molecular oxygen that so radically changed the sort of redox state of the environment that things like iron crashed out of the oceans. And at very old deposits, these are some deposits in Australia that are billions of years old, you can actually see layers of where iron crashed out of water and sort of piled up on the ocean floor. So what that means is things like nitrogen chemistry and the core things of like what does it mean to be a amino acid evolved in a reducing environment, not an oxidizing environment. The other reason that I think organisms might not do a bunch of nitrogen chemistry is because, or redox particularly, is because the redox is so complicated, right? If I imagine starting over here to nitrate and getting over here to ammonia, I mean, look at all the possible paths I can go through. Look at all the possible interactions, look at the places I can go. And if you're trying to, say, engineer specificity into a reaction, you're trying to build enzymes that navigate this set of redox reactions, it's actually quite challenging. There's a lot of possibility for incorrect reactions or incorrect product formation. So that makes it difficult. The value proposition, if you want to put it that way, for doing nitrogen chemistry or nitrogen redox is low. And then lastly, if you end up in, say, this state, an N2 state, turns out this triple bond is extraordinarily hard to break, both depending on the context. Thermodynamically, it can be difficult to break. Well, it's always, anyway, yeah. Thermodynamically, it's expensive to break, we'll put it that way. And it's also got a very high energy, a high activation energy. So it just takes a long time to break. In addition to this complexity, there's also a lot of cross-reactivity between these molecules, right? Molecular nitrogen and molecular oxygen look the same. So if you start making N2, you run into this problem of how do you tell it apart from molecular oxygen. And so what this means, because of these factors, because of evolution and the complexity of these reactions and this sort of trap that is N2, most organisms just leave nitrogen redox alone. And we basically leave nitrogen redox alone. But since we brought all this up, I feel like I have to talk about the few microorganisms, or at least some of them, that deal with nitrogen redox. And so I wanted to mention a couple of them just because they're so freaking cool. I'm actually eventually, hopefully, going to do an entire video just looking at some of these reactions and the mechanisms of them, because there's some pretty wild inorganic chemistry type things that have to happen to allow this to occur. But overall, we live down here. And most of our experience is in this space where everything's ammonia. We've got DNA and nucleic acids and urea. It's all in this ammonia state. But there are critters that will say take a nitrate and they will turn it into N2. They get energy out of this process. There are organisms that will actually take an ammonia and a nitrite and make N2. And then finally, we'll mention one other crazy branch of critters that takes N2 and turns it into ammonia. And this cycling of nitrogen between the biosphere and the atmosphere is actually a really important part of understanding ecosystem level chemistry. So I feel like as biochemists, having some grip on this is actually really helpful too. Okay, so let's just talk about a few of the crazy reactions that organisms do. One of them is denitrification. And what this involves is basically taking nitrate and making it into N2, which is actually extraordinarily thermodynamically favorable to do so. So we are going into this N2 form. This process is something they can use to get energy. Other bacteria do an anammox reaction. So in this anammox reaction, what we're doing is we're taking an ammonia and a nitrite, and we are putting it into an N2 form. So basically, we are taking this molecule here, and we are moving electrons onto this molecule here, which generates N molecular nitrogen. Also energetically favorable if you work through all these standard state reduction potentials. And the way that they extract energy from these is actually the same as we see for other metabolic things, particularly with oxygen, is they extract energy using nitrogen-based redox chains. So this is a really complicated figure. There's a lot going on. I'm not going to go into too many details, but I want to point out two things of interest. The first is down here. You might recognize this if you've looked at electron transport chains before. What this is we're basically, oops, I should zoom out a little bit. We've got cytochrome C is transferring electrons into what we're going to call complex 4. We're going to give it the name we would call it in us. And what that does is it reduces molecular oxygen to water, right? So this is the same sort of thing that happens in us. But if we go over here to this sucker, this complex, this is called the NAR complex. What is the NAR complex doing? Well, it's doing something a lot like that, something that we've kind of seen before, right? You take a ubiquinone-like molecule, so this is QH2, which is in our mitochondria, and you're dumping off its electrons and protons. But where are you putting them? Well, look at that. You're taking a nitrate and you're making a nitrite. So nitrate is acting as an acceptor for electrons coming from ubiquinone. And so, or ubiquinol, sorry. So the overall idea here then is there is chemistry that you can do that's kind of similar to what we do with oxygen chemistry, just using nitrogen in different places. This actually makes some sense, actually, because what we often will see is that something like denitrification is used by bacteria that are in an anoxic environment. So this is just doing some of the math. If you're burning nitrate, you get 350 kilojoules per mole. If you're, say, burning a fatty acid, you're going to get 650 kilojoules per mole of carbon. So this is basically a better energy source in a lot of ways because it gives you a higher energy yield. It also doesn't have the complexity of this redox chemistry going on. So basically, microorganisms prefer doing things with oxygen, but then if they need to, they can flip over and do things with nitrogen. So the problem, of course, is that putting nitrogen back into N2 is bad for most organisms, right? Once it ends up in this place here, sorry, come on, pen, there we go. We've got to figure out some way to break that N2 bond and bring it back into a state that we can use. And it turns out this is the problem, right? We've trapped nitrogen in this really stable form. But it turns out there are a couple of classes of bacteria that actually fix nitrogen. They do this chemistry here. They take an N2 and and they make ammonia. And these bacteria are cyanobacteria and rhizobacteria. Everyone else just steals from them, right? You and I can't do this reaction. We cannot pull nitrogen out of the environment to make ammonia. So we're very glad that cyanobacteria and rhizobacteria do so. They do this with this enzyme nitrogenase, which is ridiculous. Like this deserves its entire video actually, because it's so crazy. But what this enzyme does is it takes a nitrogen, N2, 8 electrons, 8 protons, 16 ATPs, and in the process generates ammonia and molecular hydrogen. So it's extraordinarily energy expensive, but for an organism that's limited in their nitrogen, this might be the thing they have to do. So they invest a bunch of energy in this ammonia formation or this nitrogen fixing of it. And so what this then does is it brings ammonia back into the biosphere so that you and I can use it rather than having to try to somehow extract and build nucleic acids out of N2 gas. Okay, so what have I said in this video? I've said that the redox states of nitrogen are very complicated, which makes it hard for organisms to deal with it. That means that most of the time, metabolism deals with reduced nitrogen. Some organisms will use, say, nitrate as a terminal electron acceptor. Others might burn NH4, in sort of the analogies here. But that's the exception. The other exception that turns out is really important for us are nitrogen-fixing organisms that will actually pull N2 out of the atmosphere and make ammonia out of it that we can use. All right, so that's what I have in this sort of high-level overview of nitrogen metabolism. In the next video, we're going to tackle what sorts of metabolic moves we make with nitrogen as humans, and then that'll help us understand nucleic acid and amino acid metabolism. So I look forward to seeing you then, and thanks for your time.